All right, so we are talking about protein synthesis or making proteins. The process is also referred to sometimes as the central dogma. We start with DNA. DNA gets transcripted or rewritten into the form of RNA, which is another nucleic acid molecule. The RNA molecule then is then translated, think of translating a language, from RNA into a protein. So taking that code that originally came from the DNA, changing it to RNA, and eventually creating a protein out of it. So in general, the two processes in making a protein, transcription and translation. Transcription, the DNA provides the code for making the mRNA. M stands for messenger, and we'll talk about what that means later. So messenger RNA, that's the transcription part. And then step 2 is the translation, where that mRNA that was just created is going to be translated into a code of amino acids. These amino acids are going to be joined together by peptide bonds. Remember, peptide means protein, and that's going to form the proteins then that are going to give us our characteristics. All right, before we move on, we need to look at the differences between DNA and RNA. So you know that DNA has the double helix. What do you notice about the RNA? Is there a double helix? No, it looks different, right? We know what the bases are in the DNA. We've talked about those, the A's, the T's, the C's, and the G's. You'll notice over here, we've got the C, we've got the G, we've got the A, but oh no, where'd the T go? We have a U instead. So there's a couple other differences, and we'll talk about those here in a second. All right, so on the back of your paper, let's just review what you know about the structure of DNA. You know that DNA stands for deoxyribonucleic acid, and the purpose of DNA is storing information that is going to be used later to make our proteins. How many strands does DNA have? It has 2. That's why it's called the double helix. These strands are referred to as the double helix, so that's the next part. DNA contains the 4 bases, A, T, C, and G, so adenine, thymine, cytosine, and guanine. And DNA also contains a sugar, specifically the deoxyribose sugar. We call that a pentose sugar, remember, because it has 5 sides, and it has phosphates. And that sugar and phosphate is what makes up the backbone. So how is that different from RNA? Well, first of all, RNA stands for something else. It stands for ribonucleic acid. It is going to carry the code from the DNA and carry that code into making the proteins itself. So it's kind of a messenger. It's a carrier, and we're going to talk about the messenger RNA later. How many strands does the RNA have? It only has 1. So it's not a double helix. It's actually considered to be a single helix. The DNA has 4 bases as well. It does have the A, G, and C, so the adenine, the guanine, and the cytosine, but we don't have thymine. Thymine is missing. Instead of thymine, we have uracil in its place. So G and C still pair up. Guanine and cytosine will still pair up. Adenine now, instead of pairing up with thymine, will pair up with uracil, and we'll see some examples of that later. RNA also has a sugar and phosphate backbone, so the phosphate part is the same. The sugar is a little different because instead of being a deoxyribose sugar, it's just a ribose sugar. So deoxy means without an oxygen. So the deoxyribose is very similar to the ribose, it's just missing an oxygen. So our ribose sugar, which is in RNA, has an extra oxygen, 1 more oxygen than the deoxyribose does. So that's where the names come from. Something a little bit different about RNA as compared to DNA is that there are actually 3 types of RNA. We only have 1 type of DNA, but we have 3 types of RNA. There's the mRNA, which stands for messenger RNA. There's rRNA, which stands for ribosomal RNA. And there's tRNA, which stands for transfer RNA. Now, the structure of these RNAs are all similar. They all follow what you just wrote down, but the difference is basically their job. Their shape is a little bit different. They're still a strand, but you'll see later that they're going to kind of bend and twist a little bit differently to give them a little bit of a different shape, and that shape is going to determine the structure that they have. All right, so now what I want you to do, you should have your entire note sheet filled out by now, and I want you to think about 3 differences between DNA and RNA, and I just want you to jot them down at the bottom. Try to do it without looking. If you need to look, you can look back. And what I want you to do, if you're working with a partner, you can come together. If you're by yourself, just come alone, but I want you to find me wherever I'm at in the classroom. Find me, come to me with no papers in your hand, and just from your memory, I want you to come to me and tell me 3 differences between DNA and RNA. Again, if you're working with a partner, you can come together. If you're by yourself, come alone. So find me and be ready to answer that question.